Okay, so today is our sixth lecture. So we have gone through sequencing data, sequence alignment, and then sequence assembly last week. So now we are going to perform the next step in sequencing data analysis, which is to call the mutation and then analyze them against the phenotype data so that you can identify genetic variants that are associated with the disease or with the phenotype of the organisms that you are interested in. Okay, so today's agenda, we're going to talk about how we call variant or mutation from sequencing data, how we annotate them, like what kind of function are they likely to be pathogenic, how do we interpret those information. And then we'll look at a few analysis techniques for linking genotypes that we detected from sequencing data to phenotypes. And those would be the quantitative trait loci, QTL analysis, and the genome-wide association study, or GWAS. So these are probably the names that you have seen in papers before. So today we're going to try to understand what they are and the analysis involved. Okay, so let's start with variant calling, or basically mutation calling. So what we know so far from previous lecture is that we have new sequencing data. We can compare them to the reference genome, or we can assemble them into contigs and scaffolds. So now we have some tools that we can use to identify differences between the genome that we are analyzing and the reference genome in the database, which might be like a healthy individual or which might be kind of related organisms. So we can identify changes that occur in our sample specifically. Okay. So there are many types of variants. So simple ones, like if you have a certain region of the genome got deleted, or there is new sequence kind of inserted into the genome, could be from kind of viral integration or gene duplication. And then sometimes you get kind of extra copies of an entire genomic region, like extra copy of genes. So that can cause problem, or it can be beneficial to the organisms, depending on whether this increase or this decrease in gene copy number, does it kind of increase the beneficial function, or does it lead to overexpression and then to the disease? And sometimes we have big changes like two chromosomes being crossed into incorrect kind of chromosome arms. So these are typically called structural variants, which are larger scale than kind of local deletion, insertion, or mutation. Okay. So usually we can call small variant kind of easier than large variant, which might be kind of counterintuitive, right? If the change is big, like it should be easier to see. But this is based on the sequencing technique that we use. We use more short-read data, and we have higher throughput. So it's easier for us to see small-scale variant. Like if there is a single point mutation, then we have a lot of reads, so we know the frequency of the mutation. If there is a small insertion deletion, then we can detect them within the limited or scope of the short reads. But if we want to detect kind of copy number variation, like we want to know if we have 2 copies or 3 or 4 or 5 copies of a gene, like if we can only sequence the gene body and they look the same, like with short read data, we have no idea if in the genome are there 5 copies, because we only see the body of the gene. If we want to know if there is a copy number variation, we need to know if the genes are in the same context. Like, are they on the same chromosome? Are they on the same location? If we can identify 5 copies of the gene with different kind of flanking region, then we know that we have 5 copies of this gene in the genome, because they are inserted at 5 different kind of locations. But otherwise, we cannot tell. Okay, so this is just to illustrate. Like if we want to be able to detect a deletion, right? We have a pair and read, the forward read and the reverse read. When we map to the reference genome, if there is a deletion, like reads that map to the middle part that's gone, like will be absent from your data. If you can identify reads that maps to this region, that means it's still in your genome. But if there is no read, that means that region is likely to be deleted. For duplication, it's the opposite. You will get more reads from certain region because it's been duplicated. So usually you get certain number of reads, now you get twice more because your genome has twice as long of the DNA fragment in that region. If there is an inversion, then this is more tricky because the sequence will flip, like from 5' to 3', it will be the reverse complement of the sequence. So if you don't have the sequencing data exactly at the junction, like where the inversion occurs, then everything will look normal. Because you thought that reads that map to the middle here that now became inverted, you thought that might be the reverse complement strand, and you can still map it back to the genome. So the only way to detect even like inversion, where you have a inverse of the 5' to 3' direction, or if you have a translocation where one genome segment merge with a different genome segment, the only way you can detect them is if you have some reads that span exactly the junction. So that's why it's difficult to see with short read data, because the chance that you will pick some DNA fragment, sequence it, maybe 150 base pair, and it falls into this region is very small. But if you have long read data, like 100,000 base pair, like the chance that it will cover this junction is very high. So if you want to identify structural variation, like you typically need to have a long read data. For copy number variation, I mentioned that if you want to identify copy number variation for a gene, which is a small segment of the genome, that's difficult. But if you are studying like a cancer or maybe some plants or animal that have variation in their copy number of the chromosome, like if it's a large scale copy number variation, like an extra copies of an entire chromosome or half of the chromosomes, then even with short read, you're going to be able to detect, because you will see many more reads coming from continuous region on the genome. So you're not going to get like more reads kind of randomly scattered throughout. You're going to get more reads specifically in every consecutive region in that part of the genome. So when you plot this kind of graph where the x-axis is a chromosome location, so these are chromosome 1, 2, 3, 4, 5, and base pair from 1 to the chromosome length. And the y-axis here is the number of reads that you have in your sample compared to a reference control. So you have, let's say you have a healthy patient where you know that his or her chromosome copy number is normal, always 2. And if you have a cancer patient and you are not sure if that person has an extra copies of some chromosome or not. So the y-axis here, you plot the read count that you get from that patient versus a healthy control. If the number of reads is much higher in the patient, then your graph is going to, your scatter plot is going to go above the middle. But if the patient has lost some DNA, maybe there is a deletion of an entire arm of a chromosome, then that's going to be a decrease in the number of reads from that region. So this kind of plot can tell us, as a glance, that if the patient has a lot of chromosome kind of duplication and chromosome loss or not, without having to do any detailed analysis, just visualizing the data. Sometimes we can see that this patient has some copies of the first part of chromosome 22. So that corresponds to these dots. Higher read count from that region of the genome. The important things why we need to be careful about calling the mutation, we might be tempted to say that anything that differs in our sample compared to the reference are all mutation. But we have to be careful because some changes or some differences are not caused by mutation. Some could be like DNA damage when you prepare the DNA sample. There can be damage to the DNA, and any damage will be sequenced into a different base, maybe. Doing the PCR amplification, there can be errors. So your final sequencing data can differ from the original starting sample. And we have also seen sequencing errors. Even Illumina, when we say that it's 0.001%, if you imagine that we are sequencing millions and billions of DNA, right, even 0.001% is going to give you some errors. So the key about variant calling is how we can distinguish true mutation from noises that can come from technical error. Okay. So first, we're going to separate this problem into two scenarios, germline or somatic variant. So germline are the mutation that we inherited from the germline cells. So this kind of occurs before the cells divide into the new organisms. So this mutation will be appearing in every cell in our body. So it's something that we have since like the first cell. So the way that we identify germline mutation is we compare DNA from our body to the reference, to other person basically. Or the best would be to compare to our parents, because anything that differ between our DNA and our parents' DNA occur in the germline cells. So this is popular for pediatrician, because if you have a disease in the child, like in the infant, like because the parents are normal, so they can kind of rule out any mutation that occur in the parent, but not in the child, shouldn't be contributing to the disease, at least not directly, because otherwise the parents would also have the same disease. So in pediatrics, they like to compare the DNA of the infant to both parents, and then look for mutations that are unique to the infant, and try to see if those can be that those mutations can explain the disease. And germline mutation is also easier to identify because they are usually either heterozygous or homozygous. So if you have 100 reads, you either gonna have all the 100 reads being the same genotype, right? If it's homozygous, or you're going to have a 50-50 being different alleles because it's a heterozygous. But for somatic mutation, which are mutations that occur during our lifetime, like different cells accumulate different mutations. So when we sequence like diseased tissue like tumor in our body, we are not gonna get like 50% mutation or 100% mutation. If we have 1 million cancer cells, like, and only 5% of them accumulate certain mutation, you're gonna have 5% allele frequency. So it's more difficult because your mutation is going to be harder to find. Usually it's low frequency, and it's not gonna be 50 or 100. It can be anything. So the estimation of the allele frequency is also another difficulty. But let's start with the easy one first. Let's start with the germline mutation first. So for germline mutation, because the allele frequency is not an issue, like it's 50 or 100%, what people focus on is to identify haplotype instead. So haplotype, I think I mentioned a little bit during the long read sequencing section, is these are mutation or variants that are not next to each other. Like they can be far apart, but during the course of evolution, the way that we pass on DNA to our offspring, these mutations go together as a unit. So these kind of mutations are genetically linked, and these are called haplotypes. So haplotype is important because sometimes you might identify this SNP, the first SNP, like C or T, but your sequencing data might miss the other two. If you don't know anything about haplotype, then you have no idea what the SNPs at the other two positions will be. But if we have knowledge about haplotype, then we know immediately that if the first SNP or mutation is C, the other two must be A and G, because these are genetically linked. They cannot be something else. They have to be A and G at least most of the time. Like nothing is absolute in biology, but most of the time we can infer that if we see a certain mutation, we can assume that the other mutation will also follow in the same organism, in the same body. So the way that we identify haplotype, which are kind of mutations that are linked together. So with short-read data, let's focus on short-read data because these are what we generally use for variant calling. So with short-read data, the only way that we can identify a haplotype that or mutation that are linked together is that they must occur on the same read, because otherwise we have no idea. So the only way we can tell that this A, T, and G are linked is that we have to see reads with A, T, and G together, like on the same read. Otherwise we have no idea. So if we have a lot of reads, like sometimes we see A and T, sometimes we see ATG, sometimes we see ATG, sometimes we see AAA. Right? So this suggests multiple possibilities. The allele A here, the first position, is mostly linked to T, but sometimes it can be found with A instead. So in this case, we can generate all the possibilities. Like if my SNPs or my mutation linked in a one possibility, right? So we saw this in the assembly lecture that if we have multiple possibilities, we can write them as graphs. So first mutation being A is always linked to C, always links to T, but they are sometimes linked to T or G at the fourth position. So we can write this out as an alternative possibility. So from our read data, our sequencing data, we can generate multiple possibilities for the haplotype, either T or G, but the rest will be ACG and AGA. So next, we need to use these information, like the possibility for the haplotype, to score, like if we have 100 reads, which of them support the first haplotype, which of them support the second haplotype. And it might not be clear because we have sequencing data in the reads as well. So some of them might match both, but with different degree. So in this case, people use a hidden Markov model, which was in the genome annotation section. So it's a model that says that we have some hidden state, which is the haplotype. Right? We know that there are some haplotype, some possible haplotype, but we don't know which one generate the sequencing data. So it's hidden. We know that these are the possibility, but we don't know which one actually occur in our patient, in our sample. We have some observation, which are the sequencing data. So we saw the read having certain mutation next to the certain mutation, next to certain mutation. So the idea of this hidden Markov model algorithm is to find the haplotype that can explain our sequencing data. So haplotype that fit the sequencing data has to be the likely haplotype in the sample, because the reads are generated from the haplotype. So if you haven't watched the hidden Markov model in the last video, go check them out. I explain it in detail what the interaction between hidden state observation and the result that we need, how they go together. But the idea is like this. So we have some possible answer, like in this case, these are the haplotype. We are not sure which one occur in the patient or in the sample, but we have some observation. And these observations, which are sequencing data, are related to the haplotype because the sequencing data should reflect the haplotype. So the rest would be which kind of statistical model or which algorithms we can use to identify the most likely haplotype. Okay. So that's for the germline variant calling. We focus on finding the haplotype, and usually that's it, because the variant is it's usually very frequent, like 50% or 100%. For somatic variant, like in cancer, for example, it's a popular example. One of the problems is the low frequency that I mentioned. But the second problem is there will be so many mutations, especially in cancer, that accumulate in the cells, in the cancer cells. And these, some of them might be the cause of cancer, which are the one that we want, but some of them might be what people call passenger mutation. So these are mutations that occur after the cancer already formed, and the cells already changed so much that many new mutations can occur. These passenger mutations are usually not interesting, because if we want to cure the patient, or if we want like a biomarkers, we want something that are causatively linked to cancer. So the variant calling pipeline for somatic mutation also include not only just the kind of mutation discovery part, but also how to filter out uninteresting mutation, basically. They want to make it so that your final result is going to be as compact as possible. Otherwise, you're going to get like a million rows of variants, and you cannot do anything because there are just too many of them. So the way that somatic variant calling pipeline work is they want you to provide as many references as possible. So for germline mutation, we talked about the parents being the best kind of control, right? Reference, basically. For tumor sample, the best control, one of the best control is the patient's own normal tissue, because anything that's in the normal tissue are probably not going to cause a cancer compared to mutations that are in the cancer cells. The second part is not only the patient's own body, because that patient can also accumulate other kind of mutations that are harmless. We can also check the genomes for other healthy individuals, like other people in the population, like the 1000 Genome Project or the Genomics Thailand project. If those normal people have some mutation frequently, right, then those mutations are unlikely to cause cancer, at least not directly, because otherwise those people will already also have the same cancer. So for somatic variant calling, there will be, if you kind of eventually analyze the data yourself, right, the normal tissue from the patient, this is kind of easy, like we know that we need it, but you will find that there will be databases of healthy individuals, and you can download that data to use as a reference to say that if my mutation falls into this group, then these are not interesting. And the technical term is called a panel of normal, PON. So you not only have normal from the same patient, you also have normal from other people in the population. And furthermore, because when we get the tissue sample from the cancer, sometimes we also get normal tissue in the biopsy as well. Right? Sometimes the biopsy is not perfect. So this pipeline will also use data from the normal tissue in order to calculate what's called a contamination. So to see that if you have 1 million cells from your tumor biopsy, what fraction of them are expected to be just normal cells. So we need this information in order to interpret the allele frequency in the data. If we have 30% normal cells in the sample, right, and we see some mutation with 30% allele frequency, like, those might be normal cells because that corresponds to the fraction of normal cells that you have in your sample. Okay, so let's think about what happens when we call variant. So somatic variant calling will use the same algorithm as germline variant. They're going to call those mutations on the tumor sample and the normal sample separately. So the first step is the same as a germline mutation. But the next step, they will take variants that were called on the tumor sample and then compare those to the normal samples, compared to the panel of normal population, and compare to the allele frequency in the normal population in order to remove mutations that are not interesting. So these are all completely separate. So the variant calling step is the same as germline. There is an additional filtering step that takes other normal healthy population as the reference. But after this filtering, like using normal people as a comparison, there's usually still too many variants for you to analyze as a human. So we still do a lot of filtering afterward, like we'll see in a few slides what the filters are. And a lot of people suggest doing variant calling with multiple tools. So with GATK pipeline that we mentioned, the software is called Mutex2 to call the somatic variant. Other software are named like Selka, GWAS. These use similar algorithms, but different parameters, or maybe different, slightly different filters. So people usually run the sequencing data through multiple software and then take the intersection. So any mutation that are called by these softwares together, like these will be more higher confident. So we are more sure that it's actual present in the tool. And nowadays, it's not beyond the scope of this lecture, but later on in this course, we're going to talk about the impact of machine learning techniques. And nowadays, people are using more and more machine learning models to help identify the variants. So we are moving away from bioinformatics to kind of machine learning based algorithms. But we still need to understand like the bioinformatics kind of principles so that we know what happened underneath. Okay. So once we call the variant, either germline or somatic, the files, the output files that you will get will be called VCF or variant call format. Bioinformatics file names are always simple. So in this file, it might look intimidating again. Right? So the data are split into two parts. The top part here with the sharp symbol. If you see sharp symbols in any kind of computer files, this usually means it's not the data, but it's a node for human to read. So it's like a README files built to the data. So these sharp symbols just kind of tells you that, okay, these files were generated according to the VCF standard format version 4.2. So it's a kind of global standard for the VCF file. It's created in 2009 in August the 5th. And this is called, the variant in this file are coded by comparing to the 1000 Genome Project, named this like 1000 Genome Pilot NCBI 36.FASTA. So basically these are the headers that allow you to yourself in the future to come back to this file and then read it and then understand what you did at the time. Also, this file will contain a lot of acronyms. Down here that I highlight with the orange bar, you see NS, like DP, AF, DB, GT, GQ, DP, right? There's so many acronyms. So VCF files help you by explaining all the acronyms at the top. So this line with the sharp symbols contain the explanation that capital N, capital S is the number of samples with data. So for each variant, if you analyze multiple samples, in how many samples that you find those variants. DP is for total depth, so this is the number of reads that map to that variant. AF is allele frequency, so it's a fraction of reads that confirm that variant. How many percent of the reads confirm that variant? So it's the frequency of that variance in the sample. So all these kind of all the acronym explained up top. Now, some of these might not sound familiar to you yet, but at least you now have some keyword to go with instead of just GQ, like GQ, GT. Okay, so after all the explanation, now comes the data. Right? So in the VCF file, each row is a mutation. So each mutation, what information are you getting? First is the chromosome where that mutation occurred. The base pair position on that chromosome. Right? Makes sense, right? If you want to know where the mutation is, you have to specify the chromosome and then the base pair position. Then if your mutation is a known mutation, it will have an ID. So this RS605 something number or micro Z1. So these are ID that correspond to known mutation in some database. So this means that this mutation is not new. It has been identified in maybe other patients by other groups in the world. But if there is no ID, this means that this might be a new mutation that you're getting. So you might have to kind of figure out how to interpret it on your own, because no one has found it before. For each mutation, they're going to provide the reference sequence. So this is what the reference genome has for that mutation. So in the reference genome is a G. In your data is an A. So that's the mutation. And if you have a more complicated mutation, so in this case, the reference is a GTC, like 3 base pair. In your data, it's either a G, so it's a deletion, or it's a GTCT, so you have an insertion. So the change, so small changes can be designated in this kind of reference and alternate column. You can compare the alternate to the reference and figure out what happened in the variant. So this variant changed from an A into a G or a T in your sample. Then this will follow by the quality score. So again, when you see quality score in sequencing data as an integer, you think about the FRET score, like the minus 10 times log base 10 of the confidence score. So the higher the better, the lower the worse. And the filter column is just a filter based on the quality score. So you see that the quality score 3 has Q10 in it, but the other rows have a pass. So pass means that the quality is good enough. But Q10 here, what does it mean? In the header here, it says that Q10 description is quality below 10. So this score 3 is below 10. So the filter column says Q10. So it's just a quality filter. So the information column, so this will tell you NS is number of sample, right? So this means that if you let's say you have 3 samples, so all 3 samples you can identify this variant. But for some variant, only 2 out of 3 that you identify the variant. So in the 3rd sample, you might not have enough sequencing data, for example. It's entirely possible that you cannot see all the mutation in all of your sample. Okay. So the info column here that gives the overall amount of information that you have. The number of samples that were found, the sequencing depth, like number of reads that map to this variant. The allele frequency of the variant overall, like 0.5. Notice that this allele frequency is not per sample, because here there are 3 samples. So this is the average allele frequency of 0.5. So it's a global, that gives you a global overview of what information you have about this variant. But if you want to know like information on each individual or each sample, those will be additional columns at the end. So these NA001 and NA002, these are 2 different samples in your data. And each of them can have different genotype. So you see here 0, a vertical bar and then 0, and the other one is 1 vertical bar and 0. So 0 mean reference allele and 1 means alternate allele. So this means your first sample, your first individual are homozygous, wild type. The second individual is heterozygous. So this 0 and 1 kind of tells you the genotype directly. And that corresponds to the format here. So you notice that the format column has 4 elements like GT, GQ, DP, HQ. And each individual, you're going to have one piece of information separated by a comma, and the second piece of information separated by a comma, and so on. So each of these information will correspond to these acronym in the format. So the first information is GT, which is the genotype, GT for genotype. GQ would be the genotype quality. So this will be the FET score again, the Q score again. How much confidence do we have that this person has this genotype? So 48 is high. But sometimes here, like second individual, for the second variant, like the data says that if he or she is heterozygous, but the quality score is only 3. So we might not be able to trust this kind of genotype assignment. So this second number is the quality score. And the third number, DP, is the read depth. So this means that the read that support that this person has a homozygous wild type, there's only one read. For the second individual, the number of reads, sequencing reads that support that this person is heterozygous, there is 8 of them. So you can kind of, either you or other software can look at this information and have some understanding, have some ideas about the confidence that we have about these mutations that we are calling. Okay. Okay. And sometimes you see a vertical bar or a division symbol for the genotype. And you will notice that sometimes you have 1 vertical bar 0 and then 0 vertical bar 1, right? So this is important if your organism or your sample is diploid, because you want to know if the mutation occur on the maternal or the paternal genomes. If the data can tell you that this variant occur on the same strand, right, occur on the same paternal or maternal genome, you will get a vertical bar. So that means that your data can tell you that all these wild type, reference, and then wild type, these are on the same molecule, right? Either paternal or maternal. But if it's a division symbol, this means that we know that this person is heterozygous, but we are not sure whether the mutation occur on the paternal or maternal strand. Copies, basically. Okay, so those are a lot of information that you can find in the VCS file. So when we want to kind of filter the variant, like to select the one that we want, the one that are likely to be linked to the disease or to the phenotype that we are studying. The bad news is it's more like an art than science. So the good news is you can just follow what people are doing. So if you go to kind of other papers or variant calling pipeline, they will have their own favorite filters, criteria. And it might look like this. This is just one example. So people might try to filter by quality score, like quality must be greater than 30, like, in order to have a high confidence about the detected variant. We might filter based on the read depth, like we want certain number of reads to trust a mutation. And there are many varieties of them. So I suggest that you can pick the paper that you like and then look at what they perform. That's only as much as I can tell. I can tell you. Okay, so this is for the variant calling part. Right? So we hopefully up to this point, we can identify all the mutation in our sample, and we can remove the one that are low confidence. And now we want to identify the one that are related to the phenotype. So that will be the annotation and the interpretation part. And the bad news is this is even more difficult than calling. But it's what we have to do, right, in order to understand the biology. So what we think about variant annotation is that we want to understand if a mutation occurs on the genome, what kind of function will it impact on the cells or on the organism. For example, if it's a mutation on protein coding gene, right, will this mutation change the protein structure? Will it change the protein function? If it's a mutation in a regulatory region, we probably want to estimate if it will affect the expression of the gene or not. So the easy thing that we can look is if it's a mutation in the coding region, like if it's a non-synonymous where the amino acid doesn't change, or if it's a if it is non-synonymous where the amino acid change, or if it's synonymous where the mutation occurs but the amino acid is the same. If it's synonymous, it's more likely to be benign. If it's non-synonymous, it changes the amino acid. Depending on which side chain change, like to different side chains, it can cause big problem, or it might be nothing. And if it's a frame shift mutation, like insertion or deletion, which change all the codon, like that can be very severe. So the nature of the mutation can give us some ideas about the severity or the likeliness to cause kind of disease. If it's a mutation on the spike side, right, it might mess with the exon combination. Like it might cause a different isoform to occur for the transcript. Like that might cause problem in the cell. If it's a mutation in the regulatory element, like for let's say in the transcription factor binding site, the mutation disrupts the binding. So now your transcription factor cannot bind to the DNA. So that can change the expression of the gene. But if it's a mutation like in the non-coding region, in the intergenic region where nothing is known, then those might be benign or we don't know. We need to study more. The second criteria is it a known mutation. If it's known, at least we have some prior study to see if this mutation, are they found in normal population or are they found in certain disease, certain patients. And there are many databases for this, like Nomad or DB-SNPs. So these databases can collect all known mutation, all known variants, so that you can, or you or bioinformatics software can go to them and then identify if the variant is common or new, for example. And then for clinical application, they want to know if these mutations have any clinical application or not. And not necessarily whether it's linked to the disease. Like sometimes we want to know if a certain mutation is linked to good treatment response. In certain cancer, having certain mutation might mean that certain drug will become more effective or less effective. So there are also databases that not only collect the kind of association between mutation and disease, but also their association with treatment response. Like PharmGKB database, it will tell you if the mutation is linked to good or bad, good or poor response to certain drug or certain treatment, so that you can make decision kind of for your patient or for your experiment kind of accordingly. So in the assignment, you get to use some of these database. So usually you will start with some mutation, like some variant. In this case, I picked a mutation in BRCA1 gene, which is linked to breast cancer. And this mutation changed nucleotide from C to T at this kind of base pair position 6207. So if you enter this information onto QTL database, it will tell you that, okay, this mutation is known, is something that people have seen before. Experts rate this as benign mutation. And there will be kind of link to the other resources, like the paper that report the mutation, the position on the genome, so that you can kind of view more information. And here, there's also the allele frequency for this mutation. So just to give you an idea that how rare or how common is the variant that you are looking at. In database like Cosmic or Catalog of Somatic Mutation in Cancer, and they will also summarize the allele frequency of each mutation separated by different cancer type. So you will know whether the mutation you found is it linked to general cancer, like it's cause any kind of cancer in any tissue, or is it specific to cancer in certain tissue. So that can give you some clue about the molecular function or mechanisms for that variant. And they will also link you to related mutation on the same gene or other study, like in this case PubMed ID, of papers that report the same mutation. Okay. So that's just many databases. I'm going to just show a few more and then we can move on. So for farm GKB, so it will tell you that this variant, which is known on CDK4 gene with certain study, there's already a study about this gene in the context of a certain drug. In this case, is somatopin, I think, maybe it's a certain some treatment. So these are clinical studies that identify that patients with some of these mutations respond well or worse to the treatment. In this case, this study contained 147 patients. So you can identify maybe candidate drug or compound that you can use to treat or that interact with the mutation. Another popular tool that many genetics labs are using is called Franklin by QTL, which is the big biotech company. So they developed machine learning model again. Like machine learning, we're going to learn about this later on, but now that we have data from many patients around the world, even though we don't yet know the mechanisms behind all the mutation, but once we have enough data, we can start to make some statistical model that can predict if we see a certain mutation, is it associated with patients from certain disease or not? Without actually knowing the mechanisms, with enough data, we can make some prediction. So this software gathers a lot of clinical data and genomics data and train models where if you put in certain mutation, if it's a mutation with known disease association or clinical impact, they will rank the severity back to you and provide you some explanation of what this mutation does, if they know, and what kind of drug is linked to this mutation. So it's like a comprehensive tool that combine all the database together. There's some certain limited use for free user, so you can try for your project. Okay. So we have all these tools that can annotate the data, like whether the mutation is known, whether it's linked to certain disease, whether it's linked to certain drugs, what kind of allele frequency is there in the population. How do we summarize all this information? And again, there is another file format called MAF or mutation annotation format. So this summarize for each row again is one mutation similar to VCF, but instead of having just the raw data of what the mutations are, MAF files will also include all the annotation. So if you run your VCF files, let's say through Franklin, and you get predicted tier of the severity, you run it through farm GKB, you get the list of predicted associated drugs. This will become columns in the MAF file. So whenever you annotate your variant, you're going to get more and more columns in this format. So in this example here, the first row here is a CORM-L6 gene, and it's on chromosome 1, this position, base pair position to this position on the plus strand. And the first mutation that we are reporting here is a missense mutation. It's a SNP, so it's a single nucleotide change. The reference allele is C, And in different tumor, different sample that we have, we identify different alternative allele. Some are wild type, some are alternative G, for example. And I cannot show the rest of the column, but this will be like, if it, so we already know it's a missense mutation. So there might be, whether it's new or unknown, there may be an ID for this. And if it's linked to drug, there will be a column with drug names, for example. Okay. So the second part of today's lecture is going to be, once we have all the genotype, we talked about how we can use online tools to annotate the genotype to understand what they are. But sometimes you are doing your own experiment, like maybe animal experiment or plant experiment. You have your own phenotype. You are not relying on the database, the clinical database. What can we do? What can we analyze with the mutation data that we just called from sequencing data? So QTL, or quantitative trait loci. So the idea is certain gene that we are interested in, there are different alleles of the genes possible. And different alleles, we expect them to have different phenotype. Some alleles might cause greater yield in the crops. Some allele might cause the animal to be larger, produce more milk, for example. In a classical genetic experiment, you will produce a lot of hybrids of these alleles. And then you will measure their phenotype. Maybe crop production in different fields or animal size, for example. So you have some trait that you measure for each individual. And for each individual, if you sequence their DNA, you're going to know if a certain individual has allele A or capital A or small a, for example, if it's a biallelic system. So you're going to have a data that one column's gonna say genotype, big A or small a. The other column is gonna say your trait, maybe the weight of the animal or the yield of the crops. We want to analyze these two columns to see if this genotype, if certain genotype are linked to greater production or maybe less to the disease or not. So when you think of having two or more genotype linked with a trait or phenotype, the statistics model that you can use, one of them is ANOVA, analysis of variance. Because ANOVA is for comparing whether different groups, in this case groups are genotype, different genotype, do they have the same average phenotype or not? Or is one of them producing better phenotype, one of them having worse phenotype? One limitation about every genetic analysis that we are going to talk from here on is if you have mutation that occur near each other, or if they are linked, like as a haplotype, you're not going to be able to distinguish which one is the cause of the phenotype or the trait, because they are linked together. In any individual that you see an A here, you see a T here. So it always goes together. We don't know if this A is the one that caused the phenotype or this T is the one that caused the phenotype. For that, you need either biological knowledge or you need to do more detailed kind of crossing experiments. But in this class, we're going to focus on what we can do computationally, which is at least we want to identify genomic region where that's linked to the phenotype. At least we want to identify the region. Which gene in the region exactly, or which mutation in the region exactly, that requires more wet lab experiment. That cannot be solved with just computational algorithm. But let's think about what ANOVA is doing for us, just in case you forgot about ANOVA. So let's say we have two alleles, two genotypes, and the x-axis here is phenotype. Let's say the height of the animal. in the example on the left, it's very clear that animals that are higher have allele B more frequently. Animals that are shorter have allele A more frequently. So if we measure the variance of the height within animal with allele A, we'll get the blue arrow. This is the variance of the animal with allele A. If we measure the variance of the height of animal with allele B, it will be the red arrow. If we combine the animal together and we don't care about their genotype, and we calculate the variance, it will be the gray arrow. It's broader. Let's compare it to the example on the right. We have 3 alleles, A, B, and C, and their heights are very similar. So if we calculate the variance for animal with allele A, B, or C, these are going to be what the variance looks like. They are overlapping, very similar. Even if we combine them together, the variance looks pretty much the same. So we already have, from this kind of visualization, we can come up with an idea. Right? If we compare the average variance between groups divided by within groups, what happens? If the variant, if the alleles, if the genotype doesn't have much effect on the phenotype, this ratio will be close to 1 because you don't get any difference when you combine the animals together and calculate the variance. But if the genotype has a lot of impact, the variance between groups is going to be much larger than the variance within groups. So this ratio is called the F-score, and it's what the analysis of variance use to identify if your sample are more to the left or to the right. Whether the genotype that you're looking at, does it affect the phenotype or does it not really affect the phenotype? So just to review ANOVA with graphical explanation. Another way that we can quantify if a genotype have some effect or not is, I would call this linear effect model. So we want to model the phenotype. In this case, maybe the yield of the crops or the height of the animal as a parameter. This is the phenotype. We want to model this phenotype as a function of genotype times the effect of the genotype and then plus some variance. This will be our model if we believe that genotype contribute to the phenotype. But our null hypothesis, our reference model, will be that phenotype is actually random. Genotype doesn't affect it at all. It's just a certain average effect size and then plus or minus some variance. So we have 2 alternative models. One where genotype plays some effect, and the other one where there is no genotype effect. If we want to support the idea that genotype is important in terms of statistics, we want to show that the alternative model that incorporates genotype information can fit or can predict the phenotype significantly better than the null model. So one way that we can say that the model needs genotype is we can look at the effect size. This is a parameter of the model. If when we run the statistics, run the kind of linear model fitting, if the effect size is 0 or close to 0, then that means the genotype doesn't really make any difference. But if the effect size is significantly different from 0, then we know the genotype has some effect. So this is another way, different than ANOVA, that can tell whether a certain factor, in this case genotype, contribute to the phenotype. And we will see this kind of linear effect model again in transcriptomics and in other omics. It will come up again and again. Okay. So some example of QTL analysis, there are many of them. So I just pick some with nice pictures. So in this example, they study the impact of the genotype in their strawberry. on the yield and on the enzymes inside the strawberry that they want over 2 years. So this kind of box plot, so this shows the result from 2 years. The first rows are the first year, and the second row are the second year. So what we want to find, what we want to see is if a certain location on the genome, if it's linked to the phenotype, right, we should see some difference in phenotype, which is the y-axis, right, when we separate the plants or the organism or the individuals based on the genotype. So in this case, genotype AA seems to be slightly producing more, I can't read the enzyme name, but anyway, and the genotype GG seems to be producing less. And this happened in both years. So this is like an external validation that this linkage between genotype and phenotype is not random. If we repeat the experiment again in the next year, we get the same result. So when I mentioned that with computational techniques, we cannot really distinguish between effects coming from mutations that are linked to each other. So if we plot like the frequency whether the two mutations will appear together in the population, certain regions will segregate together. The region in yellow, these have high degree of co-occurring. So this is like a correlation, co-occurrence. So if the area is yellow, this means that mutation that occur in this box will segregate together as a single haplotype, right? Also here. But this is red. This means that mutation that occur in this block and mutation that occur in this block segregate independently. Any individual can have different kind of mutation from both locations. Computational analysis or population level analysis that we are learning about cannot pinpoint which one is the one that are responsible for the phenotype because they are always there. So how can we calculate whether two mutations are linked or not linked? So let's say we have two loci, 1 and 2, and we have two genotypes at each position. So the total possibility will be 4 genotypes. And let's say we go to study some population and we get certain frequency. So this is a G1 is the frequency of individual with A, capital B. G2 is the frequency of individual with capital A but small b. So all these 4 must, if you sum them together, must be 1, like the entire population. The way that we can identify linkage between capital A and capital B, like if these two alleles are linked together or not, what we can compare is we look at the frequency of capital and capital B in our population, which is G1. And we compare this to the expectation. In the population, what is your expected frequency for capital A and capital B, right? If A and B are not linked, then the frequency that they will occur together in the same individual would be the same as the product of the frequency that A, capital A occur, and the probability that capital B will occur together. And this you can also compute from this table. So the linkage between certain alleles, you can calculate based on the frequency in the population. So just to look at the calculation behind this. Okay. So now we are going to expand our scope from QTL, which is specific loci, specific locus. Right. We are going to switch and expand the scale to like whole genome. So we want to analyze all loci, whether it's linked or associated with our phenotype together at once. And people call this genome-wide association study. As the name suggests, it's a genome-wide, it's an association study. So that's GWAS. And usually you see this kind of result. We'll talk about this graph later on, but this is what you usually see. Each dot is a variant, a position on the genome. So let's say you have a million variants, so you have a million point. If you have 100,000 variants, you have 100,000 points. And the height corresponds to the significance of the association. So you want to look for genes or genomic position where the graph is very high, the scatter plot is very high. There are many problems that we have to solve if we want to expand our scope from QTL to GWAS. And the first is the sheer number of loci that you need to consider. This goes back to the early papers on the number of SNPs or single nucleotide polymorphism in human genome. This is back then from 2001. Back then we already identified 1.4 million SNPs in the genome. So if you want to do GWAS and you consider the entire genome, you're going to perform essentially 1.42 million QTL. So you're doing 1.4 million tests. So this is a good chance for me to introduce the concept of p-value and multiple testing correction. So p-value, I think you are all familiar. A cutoff at 0.05 means that if there is no association, like the null hypothesis, there is a 5% chance at random that you observed an association with the score, with the same score that you have. So let's say we have 100 patients with disease X, and 40 of them have allele small a. Our null hypothesis, if there is no association between disease X and allele small a, then we expect 25% of the patient or 25 patients to have this allele. If there's no association, then you have a 25% of having double small a. We can calculate the p-value for this. We will talk about binomial distribution afterward, but we can get p-value for this, and it looks significant. But let's say we scale it up to GWAS. We are performing 1.4 million, let's say, ANOVA tests. If we extend what we know about QTL, we are doing 1.4 million QTL tests. So let's say there is no association between this disease that we are looking at or phenotype that we are looking at and 1 million SNPs. So, out of 1.4 million SNPs, we don't expect most of them to contribute to the disease. So let's say 1 million of them have no relation to the disease. If you apply a cutoff 0.05 to your p-value with no adjustment, with no correction, you will have 50,000 SNPs being significant. And these are the SNPs that have no association. So your data is going to be polluted by 50,000 false positives easily. And you know that this is a very weak estimate, right? There's going to be many more than 1 million SNPs that are not related, that are not linked. So this is a big problem. If we don't do anything to the p-value, if we don't do multiple testing correction, then your ANOVA tests are not going to give you very useful information. So that's why we need to know about p-value correction methods. And the easiest one that we are going to talk about now is Bonferroni correction. And it's simple. The inventor just tells us that, okay, you can divide your p-value threshold by the number of tests. So let's say you have 1.4 million tests, right, for 1.4 million SNPs. Your new p-value threshold is going to be 0.05 divided by 1.4 million. Why does this help? Because if we assume that there is no association for 1 million SNPs, right, assuming that this null hypothesis is true, there will be only 0.036 SNPs that will be significant by chance. So now instead of 50,000, it's less than 1. So your result, your ANOVA result that has been collected, you can trust most of all the SNPs that you found, because almost none of them are false positive. Right, so this is the importance of why we have to do multiple correction for multiple testing. And this is reflected whenever you look at this plot, which is called Manhattan plot. If you have seen any movies that about New York before it got destroyed by anything in the movie. So this looks like skyscrapers in Manhattan. You notice on the y-axis, this dashed line, which is the significance threshold, look at the number, is about 7. And this is again the minus log 10 p-value. So 7 here means that the p-value is 10 to the minus 7, which is 0.0000007. This comes after the adjustment. We are not going to apply 0.05 to the Manhattan plot, because we know that if we apply 0.05, it's going to be all the way down here, between 2 and 3. And everything is going to pass. So every time we see result from GWAS, every time you kind of feel frustrated that the p-value has to be so low, that's to prevent all the false positive that waits for you at the bottom of the graph. So in this case, the adjusted p-value threshold is 10 to the minus 7, which is very small. And any dot that goes above this threshold, these are significant associations that you can follow up on. So these genes that appear in this location are genes that might be linked to the disease or phenotype that you are looking at. In many cases, some genes or some mutations are linked. Many genes or many mutations are linked. So sometimes you know the region, but you don't know which gene in that region are associated with the disease. You only know that this region is associated. And the reason why you see many dots for each gene or for each region is there are many SNPs that are linked together. So all of them are going to show up and all of them are going to pass the ANOVA test together. So you have a lot of dots from each genomic region. Okay. So that's everything for today. So we kind of talked about how we call variant, how we call mutation. Once we know the mutation, how can we annotate them using online database. And if you are analyzing the association between genotype and phenotype, now we know the statistics models kind of underneath all these tests. Okay, so see you next session.